Okay, so let's talk about the compound light microscope. This is the microscope that you are going to be using in an anatomy and physiology lab. Normally, we talk about the microscope when we talk about histology, when you're looking at tissues, when you're studying the tissues. So let's talk about this. Microscopes are essential to the study of biology because a lot of organisms and a lot of parts of organisms are actually not visible to the naked eye. You have to use something to magnify it. They magnify things so that you can see them. And there are many types of microscopes. There really are. But the most common used ones are going to be the dissecting scope, the compound scope, and the electron microscope. You can call this the compound light microscope because it uses light. Dissecting scopes are pretty much going to be kind of like using an actual magnifying glass. They only magnify about 50 times. They're usually binocular, which means we have two places that we can look through. They also usually provide a three-dimensional image because you want to be able to see with these. Now, why am I saying you want to be able to see with them? It's because of what they're used for. Um, they usually magnify things that can be seen by the naked eye. But when we're looking at it with the naked eye, we can't see the details. So this allows us to see detail. So where do we see these used most often? Well, usually at jewelry stores or at coin vendors, because you want to see how much damage there is to a coin or how good the details on, or I guess how, if there are any impurities, I'll say it that way, in a gemstone or things like that. You can also look at plant and animal parts as far as the surface goes, but we don't really use this in A&P. We tend to use the compound light microscope. It actually uses a system of lenses to magnify up to 1,000 times. In order to provide a better image, the light actually has to be able to pass through the specimen. What that means is whatever our specimen is has to be very thin so that that light can come through it, okay? Most of the time when we're talking about specimens like tissue specimens, let's say I'm looking at cardiac muscle from the heart, you have to stain them. You have to actually put dyes in them so that you can see the different parts and pieces and you can see the details. Most of the specimens that we look at using a compound light microscope are going to be 0.25 nanometers to 1 millimeter if we're talking bacteria. Now, the third type of microscope that you will hear a lot about is the electron microscope. Electron microscopes were built in the 1940s. This was like a big, huge, ginormous deal. They were a huge breakthrough in biological science. They use beams of electrons that are focused and magnified with electromagnets. So they can either shoot these electrons through the specimen or they can bounce off the specimen. So depending on what type of electron microscope you get, whether it's an emission electron microscope or a transmission electron microscope, it'll actually give you either internal details, let's say inside of a cell, or it can actually give you what the surface looks like. Now, what's the drawback? You can't look at anything that's alive. In order for the electrons to work the way that you want them to work and give you the data that you need so that you can create an image, whatever is in this space has to be in a vacuum. So if you want to look at something that's living, electron microscopes aren't the way to go. Now, this is something that's kind of cool, though. This is why it was such a breakthrough. Electron microscopes can magnify 10,000 to 200,000 times what your naked eye can see. 10,000 to 200,000. Now, the invention of digital photos actually means that you can take that digital photo and you can actually magnify it even more, giving you something that can be 20 million times the magnification of what your naked eye can see. This is how we see viruses, for example. When we use an electron microscope, viruses are teeny, teeny, tiny. They're smaller than the smallest bacteria by hundreds of times. But with this electron microscope, we can actually see them. Even some molecules, some complex molecules like proteins, we're allowed to see with an electron microscope. So, how do we carry a microscope? Well, you've got parts of a microscope. This little thing right here that looks like an elbow, this is actually called the arm. And you're going to want to hold the arm with one hand. This down here is called the base. And again, you're going to want to hold that base with one hand. I guess I'm off the slide. So let me do that. You're going to want to hold that with one hand. It's always a two-handed kind of thing. If you don't and you trip or you lose your balance, the chance of you dropping that scope are really high. Now, depending on the scopes, they can range anywhere from $5,000 to $15,000. So when we talk about, you carrying these things, you're careful. So 2 terms you need to be familiar with: magnification and resolution. Now, if you watch the 2 videos that I've posted, you kind of already have an idea what these are. Magnification is how much we enlarge an object, okay, so that we can see it. And it's important, don't get me wrong, but it isn't as important as resolution. Resolution is kind of where it's at. Resolution, or resolving power, is the ability to distinguish between 2 points in space. Basically, you can see the small details. Technically, this image on the right-hand side of your screen, all of them are magnified to the same degree. All of them are the same size. They're all magnified. Can you see any details in the first picture? Other than 2 globy fuzzy things, probably not. This is where resolution comes into play. This has very poor resolution. It's getting a little bit better in the second picture, but by the third picture, you've got really good resolution, that ability to distinguish between 2 points. Resolution is essential for a microscope. If you've got magnification out the wazoo, but your resolution is in the toilet, it doesn't help. So resolution is really important. So let's talk about the effect of magnification on other optical properties. Okay, so as we magnify, let's talk about working distance. So if this is the objective that I'm looking at my slide looking toward my slide with, let's go with that. I'm going to draw this a little different. So here's my slide. This is my working distance. Okay. Now here's the dealio though. As I increase my magnification, as magnification increases, these objectives actually get longer, meaning that when I increase my magnification, that working distance gets smaller. The more I magnify, the closer that objective lens gets to the slide. So my working distance gets smaller. Now, that's working distance. Now let's talk about field of view. So field of view. So when you look through a microscope, you've got a certain amount of space that you see let's say your specimen in. Okay, so this space from here to here, this is your field of view. This is the view that you see of view. When you're magnifying, you're kind of wanting to get closer to your specimen, right? So let's say that this is at a 10x, 10 times what your naked eye can see, and you want to go to 20 times what your naked eye can see. When you do that and you magnify that image, basically what you're doing is you're coming in closer to this image. If you look at the distance here to here and here to here, that distance is a lot closer to those specimens. So that field of view has become a lot smaller in this example than this example. So at 10x, I'm looking at a pretty big piece of real estate. At 20x, that piece of real estate has gotten quite a bit smaller. That field of view has gotten quite a bit smaller. So as you magnify, field of view shrinks. Okay, it gets smaller. You're looking at a smaller piece of real estate, like I said. Okay. Sorry it takes so long to erase. And done. Let's talk focal plane. Okay, we've already talked about the planes of the body, right? And we said that they're kind of slicing things from top to bottom, left to right, front to back, right? In the case of a microscope, depending on how you're focusing, it literally does focus in layers. So let's say I've got a hamburger. There's the bun, here's the lettuce, here's the tomato, here's the meat, here's the other bun, okay? As my microscope focuses, it'll focus on the first layer, and then that layer goes away, and then it'll focus on the next layer. So let's say the lettuce, okay? And then that layer goes away. It literally goes from plane to plane to plane, from layer to layer to layer as it focuses. This is one of the reasons why, as stated earlier, you have to have really thin slices of specimen to look at. You can't have layers and layers of cells, or one, the light won't go through it, but two, you'll have a really hard time finding a plane to focus on. When you buy slides for the lab, you have to be very careful, because if you get slides that are really thick, you're going to have a hard time finding the landmarks that you're looking for your students to observe. So that's focal plane. Basically, the layers that you can see. Clarity. This kind of goes back to resolution. How clear is that image? Can you see details? If you have good clarity, you have good details. Okay? If you have poor clarity, then you're not really going to see anything except a big blob. So, parfocal and parcentral. One of the first things that we learn how to do when we use a microscope is to take whatever our specimen is in our field of view and put it directly in the center. Okay? That's kind of the first thing you do. First thing once you're focused, put the specimen in the center of your field of view. Now you might be saying, okay, but why? Well, it actually has to do with this word here, parcentral. In a perfect world where unicorns and rainbows were with us every single day, when you switch objectives on that revolving nose piece, when you turn that revolving nose piece and go from the 4x to the 10x, okay? When you switch that magnification from the 4x to the 10x, technically, if you've centered an object on the 4x, when you switch to this 10x, that object should still be centered. That's parcentral. It means that it's completely in the same spot. It's still centered. You can still see it where you see it. Okay? Now, parfocal, again, in the land of unicorns and rainbows, would mean that when you switch objectives from 4x to 10x, if I've put it in focus at 4x, okay, when I switch to 10x, it will still be in focus. Do we live in a perfect world? No, we don't. So while the idea is there, it's not necessarily that perfect. Now, is it close? It actually is fairly close. Normally, when you switch over, you can still see your object, but it may not be perfectly centered, so you kind of have to adjust just a little bit. Same with the focus. When you put your specimen in focus at the 4x objective, and then you switch over to the 10x, you're not going to have to move it much to get it right back into focus. In fact, you'll use your fine focus knob to put it into focus. You don't even have to touch your coarse adjustment knob because the coarse adjustment would move it way too much. So those are terms that we need to know. So again, working distance. Here's my 10x, my 40x, my 100x. Look at the distance between my objective and my slide. As I go up, this distance gets smaller and smaller and smaller. So the distance between the objective lens and the specimens shrinks. This is one of the reasons why we only use the fine adjustment knob after we get things in focus at the 4x objective. Okay? Once we have things in focus at 4x, we don't ever touch that coarse adjustment knob again. Look at how close these are to that slide. In the video, she talked about crushing the slide. If you use that coarse adjustment, it's fairly easy to crush the slide with these objectives being so close to the specimen. Again, with the field of view, if you look at how big this looks, I'm looking at it from here. So going from a low magnification to a higher magnification, I'm shrinking that space that I'm seeing through that lens. Depth of field, this is the hamburger, okay? In the lab, we actually have a slide that has 3 strands of silk thread. And depending on how they actually put those strands on the silk thread, you'll notice one come into focus, then another will come into focus, then another will come into focus. Kind of like that hamburger bun going from the bun to the lettuce to the tomato to the meat to the bottom bun. You're literally going through those planes, those layers as you magnify. Clarity, like I said, it's akin to resolution where you can actually see the details in the image. And parfocal and parcentral we just talked about, so I'm not going to go through it again. Now, let's look at the microscope and its parts and pieces. Okay, the eyepieces. In your lab on any campus at CBC, you would have two eyepieces. They're bifocal, meaning that one for each eye. But the eyepiece is what you look through. Now, normally the eyepiece is actually a magnifying lens as well, and it's normally 10x. That's just kind of the standard. Now, there may be others out there, but I'll be honest, I've worked in I don't know how many labs, and I've never seen anything that wasn't 10x. Down here is the body of the scope, okay? Then this is going to be the arm. Again, think of an elbow. You can't really mistake that for anything else because it looks like an elbow, okay? Now, if you look here, this is the stage, and this is where you are going to put your slide. So the stage actually has a stage clip right here. And that basically just holds your slide in place so that it doesn't shift around on you. Coming under these objectives, you'll see this thing right here. This is the revolving nose piece, okay? Revolving. Just like in the video, you saw that it shifted around. Back in the day, before we had revolving nose pieces, you used to either have to have a different microscope or you had to by hand take these objectives off and put a new one in place. And whether or not your scope would be in focus or anything else, pain in the rear. So we've got this revolving nose piece that actually has multiple objectives on it. Now, normally for us, for our labs, those revolving I guess so. The revolving objectives would be a 4x objective, a 10x objective, and a 40x objective. Depending on the lab, some also have a 100x objective. But you always start with that 4x, okay? So the objective lenses are these guys, okay? In the video with the cartoon, you saw the stage knobs here. They basically move your slide front and back, away from you or towards you, and side to side, okay? Left to right and front and back, anterior and posterior. One of the other challenges that you have when you're looking at something that's microscopic is if you need to shift it, if you need to shift your slide, the chances of you shifting something 1/100th of a millimeter are pretty low. So when you've got these knobs that allow you more control as to how that slide shifts, it's very, very beneficial. So again, these are going to be the stage knobs. Okay. So we've got the stage clips, the stage knobs. In the center here, you've got something called the condenser lens. So you've got this piece of glass, okay? That's not working. You've got this piece of glass, and as light comes up from the bottom, it actually focuses the light so that it condenses it, it compresses it, so that you're getting one solid beam going through your specimen. That's what the condenser's job is. On the arm of the scope down here, you'll notice that you've got two knobs. The coarse adjustment knob on the outside, which is big and kind of bulky, and then internally here, you've got that fine adjustment knob. Coarse adjustment knob moves the stage a lot, just like what you saw in the video. Fine adjustment knob moves it very, very little. Now, remember what we said about parfocal. When it comes to finding a specimen, you find it at 4x, you move to 10x, and it should be almost perfectly in focus, that's when you're going to use your fine adjustment knob. You never touch that coarse adjustment knob a second time. There's no reason to, because you're almost there. It's almost perfect. So you just need to slightly tweak, and that's what the fine adjustment knob does. If you move the coarse adjustment knob, once you've switched your objective to a higher one, to a higher magnification, you're going to run into issues and you're going to completely lose your specimen, and then you're not going to be able to find anything. At the bottom here, you have the base, okay? And then under here, you've got the light source. I don't know what that is. Under here as well, you have something called a diaphragm. You can also call it the iris diaphragm. So you know the iris of your eye, the colored part of your eye. It closes and makes the pupil very, very small when you're in a bright room, but it opens up and makes the pupil huge when you're not. That's exactly what this does. From under the stage, it'll focus this light that's coming up into a small beam, or you can open it up and spread it out, and then the light scatters a bit more. So, calculating magnification. Technically, you have two magnifying lenses. Like I said, the objective is usually going to be 10x. Then you've got your objective down here, and depending on which one you're using, that'll basically tell you what you're looking at, or how many times you're magnifying. So the big thing here is you need to know what that bottom objective is, because this will always be standard. 10x, that's what it's going to be. So let's say that I've got the 10x up here, and down here I have the 40x. Now x is the units. You can't forget to put the units. 40x means 40 times bigger than what your naked eye can see. So 10x times 40x. So 10 times 40 equals 400x. So when you combine a 10x objective up here and a 40x objective down here, the image that you are seeing is 400 times bigger than what your naked eye can see. Okay, that's calculating the magnification. If I change the objective down here to 10x instead of 40, then you would do 10x times 10x equals, what's 10 times 10? 100x. It's 100 times bigger than what your naked eye can see.